All right, let's go through this. So now we're going to do the mechanism for olfaction. All the rest of that was just a prelude to show you kind of what goes on and some of the other ways that the olfactory process connects up to other surprising parts in your brain. And yeah, here we go. So this is important for both the vision that we already learned and the olfaction that we are learning in this lesson. And they both involve complementary processes. So in the one case, in vision, we were taking a cyclic GMP and turning it into a regular GMP. Now you can do this with any nucleotide. You can take the monophosphate and turn it into the cyclic phosphate. That's fine. Cyclic phosphates like cyclic AMP are actually pretty commonly used in a variety of different processes inside the cell. Same thing with cyclic GMP. And so there's an enzyme that will make the cyclic nucleotide, and there's an enzyme that will break it back to the just regular old monophosphate that's not cyclic. So the enzyme that makes it is going to be called an adenylate cyclase or a guanylate cyclase if we're talking about GMP into cyclic GMP. But the one that you need for olfaction, we're just dealing with AMP to cyclic AMP, so it's going to be an adenylate, like adenosine cyclase. So adenylate cyclase converts adenosine monophosphate into cyclic adenosine monophosphate. And there we go, by bending that phosphate around and joining it there and popping off this OH and this H to make a water molecule. And on the other side, this is the one that we had in vision. We got that cyclic phosphodiesterase. Again, it's going to be an enzyme. The ASE tells you that it's an enzyme. And in this case, the enzyme is going to break this cyclic phosphodiester bond. This is the ester, this oxygen joined up between two other atoms. And yeah, it's a bond that makes this circular. So that is the phosphodiester bond. And yeah, we're going to break that down. So that's the idea here. Those two enzymes are at work inside the cell, but for our process, for the process of seeing something, seeing that baseball coming towards you, or that process of smelling something and then, sending that information to the rest of the brain, these are the ones that we're going to need. Cyclic GMP into GMP with cyclic phosphodiesterase, and that's going to be a GMP specific version, and adenylate cyclase doing the opposite reaction but with AMP into cyclic AMP. So this little picture right here gives you pretty much the entire test question. So I would say, yeah, this is a good one to study for the purpose of passing the test. So for smell, we're going to have a special type of G protein coupled receptor, and it's going to be called an odorant receptor, OR. And you can see that written up here, odorant receptor, meaning that it can bind to molecules that are going to give us a smell. So odorant receptors are going to be very similar molecules to the ones that were involved in vision, like rhodopsin and the photopsins. But instead of acting like a lens that focuses light down onto a particular bond in a molecule, these are just going to wait and grab onto a molecule, and it's going to be the same thing. When the alpha helices at the top grab onto that molecule, they're actually going to push out helix 5, which is attached to helix 6, and those are going to move helix 8 down here at the bottom. And yeah, basically 5 and 8 are going to change their position, and that is going to change the shape of this, which is going to activate the G protein. So the G protein for smell is going to have an alpha and a beta and a gamma subunit, like they always do. And the alpha and the gamma are going to be the ones that are membrane attached, if I remember correctly. And so this is G alpha involved in olfaction. So they still call it G alpha, little OLF next to it. And these are the G beta and G gamma. And so this is going to start out with GDP inside of it. But when the odorant receptor receives a signal, it's going to activate the G alpha subunit, which is going to get rid of its G DP and put in a GTP. So imagine that little arrow here, kind of like this, but with GTP coming in and GDP going out. And so now that we have GTP in here, it is going to go over and activate adenylate cyclase number 3. That's this thing that's AC3, which is a type 3 adenylate cyclase. And I'm saying adenylet, like an adjective, and this is adenylyl, which is another version of the adjective. It looks kind of funny with the YL there. But they both just mean a thing that makes a circle, that's the cyclase, enzyme that makes a circle out of adenosine, yeah, which is the adenylyl or adenylate. So there we go. And so this one is going to take ATP and turn it into cyclic AMP. It's not going to take just regular AMP, it's going to take ATP and turn it into that. Yeah, conceptually it's the opposite of a phosphodiesterase, but mechanically it's going to work in a different way. And now, meanwhile, cyclic AMP is also going to be one of these little keys, and it's going to open up a different ion channel. And so, once again, this is an ion channel that's going to let in a positive charge. And so, normally this is going to be shut, and so the cell is going to be, it's not going to have any positive charge inside of it. But when we activate adenylyl cyclase type 3, then we are going to start making cyclic AMP. And the cyclic AMP is then going to open up this channel, which is going to let positive charge into the cell. So in vision, we built up this negative charge, and then we fire off to the neuron, and so be it. In olfaction, we're actually going to build up a positive charge, and that is how we are going to fire off a signal to the next neuron. And from that point, it's going to work like any old neurotransmitter would, and it covers that pretty well in the book. But it's another series of ion channels that'll open up and let these positive and negative charged ions in. Now, this system works because you have a negative ion channel, a chloride ion channel, that's always open and is always, or rather, it's not always open. It opens up when you get calcium in here. So the calcium is going to open this up, and it's going to send chloride ions, Cl minus, out. So it is a calcium gated, like calcium controls the gated, chloride ion channel. And this is going to be a CAMP gated ion channel. That's what this blue one over here is. And so positive coming into the cell is going to activate this little pump, which is going to then send all the negative ions out of the cell. And so with smell, the cell is going to become super positive, and that is what is going to send the signal. We're going to send a literal positive charge signal, just like an electrical signal, to a nearby neuron, which is then going to go and signal the way neurons do all the way until it gets to the part of the brain. And yeah, it'll sort of spread out. You'll make some sort of decision, and then there we go. We got it. Now, meanwhile, there's another element here, which is the G beta and gamma subunits. So each nose cell, these are cells on a thing called the neuroepithelium, which is, epithelium is like your skin, you know, the outer layer, and neuro means it's like partially a neuron, and that's what the cells are that actually smell. These are some of the cells that can be infected with SARS coronavirus 2, the thing that causes COVID-19. And so that's why people are losing some of their smell, because they're actively getting these cells, which are neuroepithelial cells, they are getting infected, and so they are no longer signaling properly. And so, yeah, if they don't signal, you can't smell, because they're the ones with the odorant receptors. So each of these cells is going to have a variety of odorant receptors on it. But if we get a signal on a particular odorant receptor, each one is going to have its own G protein attached to it. And the beta and gamma subunits of that G protein, once the alpha subunit is activated, beta and gamma are going to go into the nucleus, and they're actually going to shut down production of the other odorant receptors that this cell could make. And so in this way, if you smelled one thing, you'll send a signal, you'll send off a little positive charge, send out some negative, signal to a neuron, but you'll also lower the amount of other receptors that you make. And so you're going to be ready to smell the same thing again. And if you've ever noticed that, you smell one particular person or one particular thing, and then over time you just get more and more sensitive to it, That's exactly what's happening. Your nose, in those neuroepithelial cells, your G, beta, and gamma subunits are actually blocking transcription of some of the genes for the other odorant receptors so that you can smell this thing more. Whether you want to or not, that's the thing that your neuroepithelium is doing for you. So that's the idea. That is the mechanism. And I will make another video where I diagram this out on the whiteboard, but it's going to look pretty much like this. And yeah, that's the challenge. This is the thing to learn. So you're going to maybe want to watch this a couple times, take notes on what's going on, look at all the lovely text that's over here, try and sketch it out or try and write it out in steps in your mind. And yeah, once you've got all the steps down, then that's going to be a pretty good answer to my test. So thanks very much. That's the end of this part.